Hello students, this is the platform of distance e-learning and I am your science teacher Sulochana. Today we are going to talk about DNA replication. How DNA replicates, right? So as you know that we are having double-stranded DNA. Double-stranded DNA means that we are having two strands. Now if you see your DNA is like this, fine. So it is complementary strand, means if it is starting with 3' end, it will end with 5', and if it is starting with 5' end, means that end is open. What do you mean by this? 3' end means your OH group, that is functional group, is free here. If you talk about 5' end, so here phosphate group is free, right? So this is how we are having a double-stranded DNA. Now, when your DNA has to replicate, how this process occurs, right? So you all know that we are having nucleotides in our DNA, right? That is adenine, thymine, cytosine and guanine, right? So these are in the inactive form in your cell, right? It is not like this that every time they are active, because if they are active, if they are found in active form, then they will keep on replicating, right? But every time your DNA need not to replicate. So in your cells, it is found in inactive form. So it is like in the form of monophosphate, right? Adenine monophosphate. Then it converts into ATP. Adenine will convert into ATP, right? Adenosine triphosphate. Here it is TMP. It will convert into TTP, means triphosphates it will start converting, right? CMP, that is your cytosine, it will convert into cytosine triphosphate, again GMP, guanine, right, monophosphate, it will convert into guanine triphosphate. Now when they will come in this form, this is the activated form. This form is activated, right? Your DNA is ready to replicate now, fine. Now all the nucleotide which was in inactive form, it is converted into its active form, that is into triphosphates. Now what has to be done is, your double helical DNA, it is like this coiled structure. Now it has to open, right? It has to open. Now it will open, which will help it. So when it will open, first of all, it has to recognize that from where DNA has to replicate, right? So the site from where DNA will start replicating, it is called as Ori site. Getting? So first of all, the Ori site has to be recognized, right? After that, then it will start what? Opening. So helix has to open. So helicase will help the helical structure, this DNA, to open. Now, if you imagine that you are having a coiled rope, right? When you will open it, again it will get coiled. It happens. Now you want that both the coil, it should be, means both strands should be on their own space, on their own position. So how will it occurs, right? So which will help it out. So it is topoisomerase and single-stranded stabilizing proteins which will help the strands to be on their position. It will help in releasing that tension which helps the double-strand DNA to coil again, right? So topoisomerase, it keeps the strands in its position, right, by removing the tension in the strands. Are you getting? So now your both helix are opened. Now primer has to come and attach on the Ori site from where replication has to start. Are you getting? So primer has to go there. So here next is DNA directed RNA polymerase. It will help in making the primer. Now after that, DNA replication will start. Fine. So DNA replication, it always move, the new strand will always come from 5' to 3' end. Getting? So 3' end is here, functional group is here. Always remember that primer will attach on the 3' end, right? Wherever is the 3' end and the Ori site is recognized, it will start replicate, the primer will attach over 3' end. So your primer is here. Now the next nucleotide has to come here. Fine. Then the next nucleotide, then the next nucleotide. So nucleotides start attaching. Getting? Now if you talk about the next strand, so it is from 5 prime. As you know that primer has to come on the 3rd prime end. So primer will come here, right? So from here, then again the nucleotides, they will start attaching. Are you getting? Now if you see, so when the chain comes here, it will terminate, right? Because now it is not getting the complementary nucleotide because it is end up here, right? So what will happen? It will lag behind, you know, and this will become your leading strand. It will keep on opening and the nucleotides will keep on attaching. But we have to make the complementary strand for this strand also. So what will happen? From here, then it terminates. Again, the primer has to come on the 3 prime ends. Now imagine the 3 prime end is here. So primer will attach here. Now further the nucleotides they start attaching, right? So similarly what happened? That the both strand, if you see both strands, so one strand will be your leading strand. One strand will be your leading strand because here the nucleotides, they are not having any gap in between, right? So it is leading. And if you see the next, so you are seeing the Okazaki fragments over here. Okazaki fragments means that this strand here, the primer has to attach again and again, and then the complete chain was here. Now you see that fragments are here, right? 1 fragment, 2, 3, right? So fragment is there. So now at last, now here you see both the complementary strands are having the other strand along with them. So from here, now we want that these Okazaki fragments has to attach. So which enzyme, which protein will help here? So it will be ligase, right? So ligase will help in attaching all these Okazaki fragments, and this is how your DNA replicates, right? Now here I'll like to tell you that in prokaryotes, mostly we are having one this site, right? But if you talk about the eukaryotic cells like us, right? So we are having, sometimes we are having many sites. So there the replication is little typical, but otherwise if it is one site, it occurs like this. So it is easy for you to understand. So I hope you all understand this concept, DNA replication. Very, very important topic and very easy to understand. It starts with first your inactive nucleotides, they get activated, right? Then second point, opening of DNA helix. Helicase will help in opening, and topoisomerase, it will help in removing the tension, right? Then the primer goes and attach on the 3 prime end of the both strands. So then you see that one will be your leading strand, right? And the other, you feel that Okazaki fragments are present over there, and these Okazaki fragments get attached with each other with the help of ligase, and this is how your double-stranded DNA replicates. This is all in this topic. Thank you. I hope you like the session and you understand it properly. Please, if you like the session, give me a thumbs up. Do comment so that it motivates us also. So do comment. Thank you. Have a nice day.